The world as we know it has changed. We are sitting in the post pandemic era, an era which is driven by science and scientists. Thankfully, today we live in an era which is propelled and accelerated by science. 120 years ago, it was not even possible by humans to fly in the sky. But today we are editing genes. We are slated to grow bigger and better. Now, as rightly said by Team Merck before this meeting to me, that science is what should drive the economy of the country. Science is what should be the paramount engine of our country. And in that direction, today I am pleased to introduce Dr. Debojyoti, who is the recipient of MERCK Young Scientist Award 2022. And he's joining us from his lab right now. And I'm so happy to inform all of you that MERCK has facilitated this interaction. And I'm thankful to the communications team at MERCK. And let me bring up Dr. Debojyoti to the stage now. Hi, Dr. Debojyoti, how are you?
Hi. Thank you very much for having me here.
Thank you so much for joining us. I know you have a busy schedule at your lab. So quickly, let's jump in and understand what are you working and on right now, and how are things at your end?
Yeah, so my major focus from my lab is actually on CRISPR-Cas based genome editing and different applications, majorly in therapeutics and clinical applications. We also did some work on diagnostics using CRISPR at some point. But the main area which we are currently working on is trying to bring these gene editing based tools for actual correction of diseases in humans, which is in the patient population in India, starting with sickle cell anemia and then with some other diseases.
Right. So I can see. So I was reading your LinkedIn profile, and a lot of accomplishments which you already have before achieving this award. So if you could throw some light and share your journey of transitioning from studying non-coding RNA in embryonic stem cells, and then how it went on to establishing a research group focusing on CRISPR, how the journey has been, and what all challenges which you faced being from our revered group now. So please go ahead and share your journey with us.
Yeah, so I, of course, when I was doing my PhD and my postdoc, the work that I did then, and a part of it which we continued also after I became a group leader, was principally at the crossroads of embryonic stem cell pluripotency, differentiation, and so on. When I joined as a group leader at IGIB in 2016, I actually made a conscious decision to change my field because CRISPR was coming up in a very big way at that point, and I kind of felt that joining IGIB, being in Delhi, being in a CSR institute, gives a lot of unique opportunities for translational work. So I made this conscious jump into gene editing, probably not with the thought process at that point of time to really make a clinical application, but it kind of turned out into that way. So I always had the let's say the dream to be able to have some tangible outcomes from the type of biological research that we are doing, how to take people, and I think that gave me this opportunity. CRISPR-Cas gave me that opportunity to do that.
Wonderful. So you are a recipient of MERCK Young Scientist Award 2022. And how does it feel getting acknowledged for your work and this award?
It feels great actually because I think awards, if anything, they are a big motivation. Means that your work is getting noticed. It also means that your work is coming under a lot of scrutiny. So it comes with its own responsibility as well. So that if you have set a bar which is being noticed and acknowledged, there is much more that is expected from you. Which is what the Merck. So I was very fortunate to actually have two Merck Scientist Awards, one in the biological sciences and then one for COVID research. So that was a kind of a lucky break for me, I would say. And I'm thankful to Mark for giving this kind of an award. But yes, I think more than anything, it is a motivational achievement for me to be able to do something which deserves this kind of an award.
Right. So, Dr. Devjyoti, as you see, post COVID, the world is no longer the same. We all agree to that. But how do you see, as a scientist, the growth in the scientific, especially biological sciences and chemical sciences? How do you see this going forward from 2023 to, say, 2030, in the next 7 years? How do you foresee things?
For one, I think COVID has definitely brought out the scientist in a lot of people, actually. So I think things which are scientific terms and scientific concepts, which one a layman would never be actually discussing, has become common after COVID, because they are aware of things such as, even quantitative PCR and remdesivir and vaccines and so on. So it has done its bit in terms of educating scientifically. Now, whether or not it will translate into more active involvement in R&D funding is what we are all hoping and expecting, because it clearly showed that, when a pandemic like this hits, the only thing that can really, almost, one can use the word, protect a population, is having a scientific understanding of the disease and its therapies. So one of the pertinent problems forever in our country has been the lack or the dearth or the reduction in scientific funding at different levels. If the COVID pandemic can, really motivate everybody, whether it is industries or the government or whoever it might be, philanthropic, to put in more money into research, which can give rise to tangible outcomes, I think that would be the direction in which I'm seeing the next decade going. That there would be a lot of good work that might come because of this renewed energy or renewed interest in science.
Hmm. That's really inspiring. And so this brings me to the next question, actually, about inspiration. So now that you are diverted towards CRISPRs, what really inspired you to start a research group under IGIB for CRISPR in 2016?
Like I said, that when I was finishing my PhD and postdoc, the CRISPR field was just blooming, and we didn't have any therapy in the market yet. But there were people quite actively working on CRISPR. We, in our lab, in a very small way, had started looking at these CRISPR based gene editing applications. When I came to IGIB, like I said, I consciously proposed that I'm going to work on this and look at ways by which CRISPR can be used to manipulate DNA, RNA, and so on. And then we struck across an orthogonal Cas protein from a bacteria called Francisella cerevisiae, and then we started studying it in depth, and we found very fascinating features about this protein, which not only was helpful for diagnosis but also for genome editing right now. So these things we learned over time, and that kind of continued or built the trajectory for my lab.
Wonderful. So I'm also told that you got a chance to meet Professor Dr. Jennifer Doudna at the RNA meeting. How did that influence your decision?
That was public domain knowledge. I didn't know that. Anyway, yeah. So I had actually. It was a very remarkable meeting, because I mean, of course, before Dr. Doudna, I had a chance to meet several Nobel laureates in the past as a member of the Indian Lindau Nobel laureate meeting contingent when I was in my undergrad studies. But meeting Dr. Doudna was a bit special, because I remember it was an afternoon at EMBL, European Molecular Biology Lab in Heidelberg, where she was supposed to speak. And then this was in 2014-15, and she had not even won the Nobel Prize then, 2015 to be exact. And there was already a kind of hush in the air that this talk is going to be there at 4 o'clock. So get ready and grab your coffee, get into the auditorium, and just sit over there. And then when the talk started, it was actually, there was no space for a single person to sit in this auditorium. So much so that even the stairs were full with people. They were sitting on the stairs. And then it was, of course, a fascinating talk, like she always gives, very inspiring. And later on, I managed to grab her for coffee for about 15-20 minutes, discussing about our projects and so on. I dare say that that was one of the inspirations behind actually coming into CRISPR, because just the way that she showed the applications, and the extent to which CRISPR can go in solving a lot of humanity's problems, was one of the reasons probably why I really decided to take this up as an area to work on as an independent PI.
Wonderful, wonderful. That was really exhilarating experience. Coming to now that you are working on CRISPR, how does CRISPR technology fit in, and how can it revolutionize the any sector wherever you are researching in right now? What are your thoughts?
Lot of opportunities, but lot of challenges. First of all, I think the most impactful area is, of course, in healthcare, because CRISPR can offer solutions to some genetic diseases where there are absolutely no drugs that are available. Because when you change something at the DNA, you can essentially cure a disease. And things that we are working on, for example, for sickle cell anemia, or for some rare diseases, for the eye or the brain, um, perhaps CRISPR can actually give an opportunity to cure diseases completely. So that's definitely there. In agriculture, sorry, it can be actually used for improving traits and giving rise to better crops with higher yield. CRISPR diagnostics we have already explored during the pandemic. We know what are the advantages and limitations of that. But even for simple things like basic applications in microbiology, for food processing, for industries which are just trying to enhance their production of certain types of chemicals or biomolecules or biosimilars, whatever might that be, CRISPR comes in everywhere. So in fact, it is so much so that it's very hard to find somebody who doesn't know about CRISPR these days, or isn't using it in some aspect, in different ways. Of course, yeah, if I just concluded saying that there are huge challenges also, because there are safety challenges, there are regulatory challenges, and most importantly, there is also the challenge of the cost, the price to the patient, whether it is a first world solution or a third world solution, or a developing nation versus a developed nation. All these have to be seen. So that's why we have multiple committees and boards which are now actively discussing this. Some of them also I'm a part of now, both at the level of the government as well as at a more private level as well.
Right. So this brings me to two questions actually, which I'm as an entrepreneur myself very curious. So one is, so right now CRISPR is at lab scale, and obviously there are ethical issues, regulatory issues, and all those issues. It's always there. But how do you foresee CRISPR entering the industrial domain, and in future can be commercialized? Which particular will domain will get commercialized first, and what is your thoughts on that?
I think the areas of both healthcare and plant biotechnology would be probably one of the first, and the other one is, of course, the bioprocess manufacturing, because all of them used some kind of a biological entity at some point, where enhancement of traits, or let's say optimized traits, would actually be very useful for the final product. Healthcare, I think the first thing would be probably healthcare, because we have seen the success of CAR T, and the way CRISPR can come into CAR T, maybe HIV correction, sickle cell correction, like we are attempting, and so on. All these would probably be the areas where CRISPR comes in very beautifully. So yes, for industries, I think healthcare and agriculture would be one of the first areas.
Right. So now that you're yourself a young MERCK scientist recipient, but my question to you will be, when you meet young scientists or aspiring students who are planning to become scientists just like you, what would be your message, and what, because the research scenario in India, and there is a lot of hustle required. So what would be your suggestion or message to them?
I think you already captured my suggestion once when you said that the research scenario requires a lot of hustle. I think that is really the main thing with any kind of research which you want to move to an application. It cannot be probably done in isolation, sitting in a lab, in an institute, in some corner of the country. You have to move, you need to collaborate, you need to, in a way, sell your science to the right kind of people, whether they are entrepreneurs or government or funders or philanthropists or whoever it might be. And in a way, scientists of these days have to wear different kinds of hats, not only that of an academician but also of a businessman, kind of. So my suggestion would be that, come into an academic scientist position only if you are really thinking of having these kind of these you're comfortable with such roles, because that's important to probably stick around with the issues that we face with funding and demotivation with publications, etc., etc. So you need to have that conviction, and also the never say die attitude, kind of, that it will probably in the end lead up to something or the other.
Right. So that was quite insightful. I'm sure the young audience will, would love to listen to you once again in the future. Probably we can catch up. But now that CRISPR is what you are researching on, and worldwide we have seen a lot of criticism about playing God, right? And when you will probably, I'm sure you must be meeting policymakers, you must be meeting a lot of regulatory, bureaucratic people. How would you convince them to release funds for, something as controversial as CRISPR, as the way people take it nowadays? How do you deal with that?
If I had to answer this honestly, I have never had any problems or issues with funders. I think the funders, but even in India, are particularly interested in actually getting this field to develop in India. They are super positive, super confident, and there is a fair share of funding which has been put already in CRISPR research. Um, Policymakers, they are still sitting on the fence a bit. They do not know, because it's something that is kind of debated worldwide. So I must say that, this the concept of playing God is something which was actually, how to say, invented by the media a bit, kind of as an overstatement. Because to be very honest, with 4 billion almost letters in the DNA, and almost we knowing only 1% of how the genome functions, probably there is no way one can actually play God simply by changing a few letters. So that's very, very far fetched. What you can definitely do is that you can actually make changes in a heritable manner. Now, whether these changes are safe, what are the ethics surrounding it, for when and where they should be done, should be the topic of discussion more and more, rather than saying that humanity can now be controlled in terms of its blueprint. I don't think so that will happen in the near future at all.
Mm-hmm, mm-hmm. Great. So now that you're a vibrant part of biotech industry doing research on CRISPR, how do you keep up with the latest in the biotech industry? Like, how does your day go about it? Like updating your knowledge with the current trends. How do you do that?
I fall short. I mean, I am probably falling short by several days, months, or years. I think it's not possible just to keep up. One good thing that I have seen is by social media has actually helped a lot. Is that several of the scientists and fellow colleagues and students and biopharma industries are on Twitter, and we, in a very crisp way, you can more or less get the gist of very important discoveries, for which you don't earlier you'd probably have to search on a search platform, get the paper out, read, etc., etc., which you do not need now. So that's one way. And the other one are people and hosts like you, who are actually highlighting important discoveries and important people in the industry, and we get these news feeds all the time, I think, from even for Biopatrika as well. So that also helps to keep abreast with what is the knowledge of the know-how, particularly in India, and sometimes also from the rest of the world.
Wonderful, wonderful. Thanks for mentioning Biotechnica, by the way. So now I'm told also that you are also an educator, mentor, as well as a researcher. So how do you balance your life and playing all these roles every day?
Well, I'm blessed to have a very good group of students. I think it's a pretty big lab right now, and and I'm myself quite young, I would say, within the academic lineage. So I would not say that I'm a great mentor or even a mentor for that matter. I'm still learning on the job, and whatever good things that I have learned from my supervisors, my mentors over the years, I try to pass in. And then there is this culture of seniority, who train the juniors, and juniors who train the trainees, and trainees who train the train their whatever. So that way, I think this mentorship process goes on, and then everybody develops one kind of style which works for them, not necessarily overlapping with other people, but that's how it works.
Okay. So one last question. You have a lot of experience of Indian academic research. So if you have to change or probably look for a change in one thing in the Indian academic research scenario, what would be that thing?
Um, okay, that's a tough question. I think the academic scenario in is very different between government versus private institutes, mainly with regards to some of the bureaucratic processes. So, if the bureaucratic processes for purchase and stuff like that, these can be maybe a little bit easier. That would be good. The other thing is that I feel that we need to have a more scientific mindset in most of these discussions and events and so on. That automatically makes the entire generation very inquisitive. So those are some of the things that I feel can be changed. I mean, there are a lot of technical small things, nitty gritties, which can be changed to make things better. But I think overall, if you had to say that for a generation, this would be probably, I think, what would matter.
Very true, and rightly said, aptly said. I must mention that we have to obviously evolve if we want to grow. And thank you so much for your time today, Dr. Debojyoti. It was really insightful talking to you. We will definitely meet whenever I'm in Delhi and I'm by the side of IGIB. But for now, thank you so much, and congratulations once again on receiving the MERCK Scientist Award 2022.
Thank you so much. I would definitely like to meet you as well and discuss this in person.
Thank you. Thank you so much.